Hi, this is Dr. B, and this is part 3 of the Enzymes Lab in Gizmos. And we're looking at Claire, a dog who has problems with her digestion. She's not absorbing the nutrients in her food, and she's lost a lot of weight. There are 3 problems we think she has: an enzyme active site mutation. When a cell makes an enzyme, one of the amino acids can be changed to a different amino acid. This is called a mutation and can change the shape of the enzyme's active site. This little yellow part right here is the active site. If it's mutated, you can see that the enzyme's active site changes shape, and the substrate, what the enzyme acts on or reacts with, does not fit here anymore, and the enzyme doesn't work. So that's one possibility for Claire. We can just do the animation here. Abnormal pH is another option. The enzyme shape depends on the pH, the acidity of the environment. If Claire's body was not keeping her small intestine at the correct pH, none of her enzymes would work properly. So let's see what that looks like. At normal pH, things fit fine. Her enzyme denatures or changes shape at the wrong pH. Remember, enzymes are very fussy little fussy little proteins. They have to have exactly the right temperature and exactly the right pH to function properly. An enzyme can only act on one molecule at a time. This means that the reaction rate depends on the concentration of an enzyme. If Claire makes enzymes too slowly, the enzyme concentration in her small intestine will be low. So here's low enzyme concentration, here's a higher enzyme concentration. This isn't very efficient, where this is far more efficient. Think of like checkout lines at the grocery store. If there's only 2 cashiers, it might be a really long time before you get checked out. If there are several cashiers who are working the checkout line, things will probably go faster. So you can see there's a lot more product. The substrate is broken down a lot more quickly in this situation where there are more enzymes. So let's perform 2 more experiments to help you make your hypothesis. Let's repeat the experiments to measure the breakdown of protein by liquid from the small intestine. This time we'll take 1 milliliter of liquid and add it to the substrate at pH 9, which is the normal pH of the small intestine. This graph shows the control data of a healthy dog breaking down substrate at pH 9. Now here's Claire's data. And remember, if we're breaking down substrate, the concentration of substrate should go down. So the healthy dog is breaking those proteins or whatever they're supposed to be breaking down, proteins, carbohydrates, or lipids down efficiently here. Claire is not breaking them down very efficiently. Her line is more shallow. It doesn't have the steep slope that this one does. We have taken 1 milligram of pure protease from the small intestine liquid. Add the 1 milligram of protease from the healthy dog to the substrate. Again, the experiment will be run at a pH of 9. So here's a healthy dog, and here's Claire. Now, it's important to understand the difference between these 2 graphs and these 2 experiments. This is 1 milliliter of liquid from the stomach. So 1 milliliter of healthy dog liquid versus 1 milliliter of Claire's liquid. Here, they purified the protease, the enzyme, so they have 1 milliliter of enzyme, so it's a more concentrated situation, and they're both the same. So Claire, her enzyme is working properly, but it looks like maybe there's not enough. If Claire didn't have enough enzyme, it'd be dilute. The concentration would not be very much. It would be low. It's like having Kool-Aid powder or lemonade powder that's too dilute. It's not enough powder and it doesn't have a lot of flavor. So it's dilute, it doesn't have enough enzyme. These are both pure enzyme, and so that's working just fine. So that can lead to our hypothesis, which is what we're going to do right now. Using the data, choose one of the 3 possible problems below. Enzyme active site mutation, abnormal pH, or low enzyme production. Let's just go with the first one and see how that goes. Active site mutation would mean Claire is not digesting her food at all with that particular enzyme, which in this case is a protease. Protease is an enzyme that breaks down protein. The beginning of the word pro means it breaks down protein, and ase tells you it's an enzyme. To test your hypothesis, you will perform 3 experiments. 1 experiment for each possible enzyme problem. Active site mutation, abnormal pH, and low enzyme production. Let's start our first experiment here. Let's examine the binding of substrates to the active site of Claire's enzymes. So we're going to collect a sample here. Now let's analyze our sample. A lot of things that they do in gizmos are kind of magic. You don't really do this in the lab. You can't see an enzyme. It doesn't, even under a microscope, it's too small to see even under a microscope. So you have to make some inferences from other kinds of tests. So we know that this is a lipid because it's like a rectangle that fits in the rectangle active site of this lipase. Lipase also gives you a clue because it's LIP, and it's LIP for lipid. Let's look at the carbohydrates here. This has kind of a little house shape or a pentagon, an open pentagon. And if we look at that, it's a carbohydrate. So carbohydrate is the substrate of carbohydrates. This is the enzyme, this is the substrate. The substrate is the thing that fits into the enzyme. And that leaves us protein. It's got a little triangular tip here that fits into the protease, or the enzyme that breaks down protein. So we're going to look at binding between the substrate and the enzyme for both carbohydrates, proteases, and lipase. We wouldn't be able to see this in a real lab. We'd have to do other tests and infer from those tests what was going on in our test tubes or Eppendorf tubes or whatever we were looking for. I'm going to say all enzymes fit their substrates so it is unlikely that the problem is an active site mutation. Enzymes work best with a specific pH range. This is called optimal or normal pH range. Remember, enzymes are very fussy little proteins, and they have to have just the right pH and just the right temperature to work properly. To test the effect of pH on Claire's enzymes, you need to examine the reaction rates on her enzymes at different pH levels. So let's look at what's happening. At optimal pH, at low pH, the enzyme is the wrong shape and the substrate doesn't fit, so it doesn't work. At optimal pH, the substrate fits and it gets broken down by the enzyme because the enzyme is the correct shape. At high pH, again, the enzyme changes shape, the active site isn't the right shape anymore, and the substrate doesn't fit. So that's why pH is very important for enzymes, because if they're not at the right pH, they change their shape and then the substrates don't fit. Let's begin by investigating the reaction rate of Claire's protease enzyme at pH 6. To do this, you'll add 1 milligram sample of protease to 1 milligram of protein substrate at pH 6. So here's the time in seconds, here's the product, and so the reaction rate here, we're going to remember how to do this. It's product made over time. So at 3 seconds here, we're at like 1.5 milligrams. So it's 1.5 divided by 3. So 1.5 divided by 3 is 0.5. At pH 6, the reaction rate was 0.5 milligrams per second. We'll plot this on a second graph. Select the dot that shows a reaction rate of 0.5 milligrams per second at pH 6. So here's pH 6 and here's 0.5. Let's start the reaction rate for pH 7. The line here for pH 7 is much steeper. So the reaction rate was higher at pH 7. Now run the experiment at pH 8, 9, 10, 11 and check the pH to find where the reaction rate for that pH. So we're basically checking like where the sweet spot is for this enzyme for pH. So it looks like the best reaction rate is here at 9. So it looks like the protease, the enzyme that breaks down protein, the carbohydrates, the enzyme that breaks down carbohydrates, and the lipase all have a sweet spot at around pH 9, which is where the enzyme works at 90% of its maximum reaction rate or more. So let's measure Claire's pH. Is the pH of Claire's small intestine below, within, or above the optimal range of each enzyme? Well, we decided the sweet spot was around 9 for all 3 enzymes, and Claire's at 9.1, so she's within normal for all 3 enzymes. So let's revise our hypothesis. This is not a very good hypothesis. Don't use this one. And we should have rewritten it for our for pH 9. So we could have said, if Claire has abnormal pH, then she will, and filled that in. Right now, we have to revise our hypothesis because it's not, we know it's not abnormal pH. Claire is right within kind of the sweet spot here, so it's probably low enzyme production. We could say if Claire has low enzyme production, she will, and you can fill in the blank. Remember, it's kind of like if she's having low enzyme production, it's kind of like only having a huge crowd and only one person running the checkout line, which is not very efficient. It's more efficient to have a bunch of people checking out shoppers. So if she has low enzymes, she's not going to be digesting very efficiently. And we'll see a low rate. So let's go to our final experiment here. If Claire produced low amounts of protease, lipase, and carbohydrates, she would not have enough enzymes to quickly break down her food. These enzymes are produced by the pancreas. We're going to collect a sample from her pancreas. Now let's analyze that sample. So these are the healthy dog comparisons here. And here's Claire. So the healthy dog is digesting at a much faster rate, making protease at a much faster rate, making carbohydrates at a faster rate, and making lipase at a faster rate. Claire has a very low rate of enzyme production for all three enzymes. So she's low for protease, low for lipase, and low for carbohydrates. Now we have some data to support our hypothesis. So we could say the data that supports our third revised hypothesis is, and then you can figure out what that is. Look at the graphs and make a decision. The enzyme production experiment showed that Claire was not producing enough enzymes. Low enzyme production is caused by a condition called exocrine pancreatic insufficiency. It happens when the cells of the pancreas become damaged, stopping the pancreas from secreting enough digestive enzymes. Here's the sample amount of protease from either dog gave the same reaction rate. If Claire's protease had an active site mutation, then the reaction rate of her protease would have been lower. This rules out an active site mutation as a possibility. Remember, when you took pure protease, things were fine. But if you just took liquid, 1 milliliter of liquid from both dogs, Claire's digestion was slow. If Claire's small intestine had a normal pH, then Claire's protease would not work properly. In graph 2, the experiment was run at normal pH of the small intestine. At pH 9, Claire's protease would return to normal and the reaction rate should be the same as a healthy dog. But it wasn't, so it's not the wrong pH. Only low enzyme could be the problem. The solution was to give Claire supplemental enzymes. So they just put some medication in her food every day. They give her enzymes that she's not making, and she's then very healthy. Adding enzymes to supplements to Claire's food will increase the enzyme concentration in her small intestine, and this will help her digest food and gain weight. We're going to give her some extra enzymes here. They're going to go to her small intestine, and they're going to help her break down her food better. So she's getting extra enzymes externally, like they're added to her food. After 2 months, Claire's a happier dog. She's playing, she's gained some weight. So we were successful in treating our patient here. And so now it's time for you to do your case summary. Describe Claire's symptoms, explain how they were related to metabolism and homeostasis. How did Claire's digestion rates tell you that the problem was in her small intestines and not in her stomach? Well, we tested her stomach pH and her stomach enzymes, and it was all fine. We determined Claire had low enzyme production because we worked through the other options, and we found that she didn't have the same concentration of enzymes as a healthy dog, so her rate was lower, her rate of breaking down things. We also know that her enzyme was okay. It wasn't an active site mutation, because when we got just a pure milliliter of enzyme and not a milliliter of stomach fluid, things were okay. She just had a low concentration of enzyme. How does Claire's diagnosis explain my 1 milliliter of liquid from her small intestine digested protein slowly? Well, that's just liquid and a bunch of it's small intestine liquid. So it might be food, it might be enzyme, it might be a lot of different things, or water that she's drunk. 1 milliliter of pure protease is they concentrated a bunch of milliliters of liquid from her small intestine and got just a milligram of pure protease. And that was working fine. The data that said her protease was working fine gave us the information we needed to know that she just didn't have enough protease. It was a dilute solution of protease in the liquid from her small intestine. So thanks for watching. Good luck on the rest of your assignment. I'll see you in our next case.